we're going to be looking at and preparing for your upcoming lesson on DNA and RNA. And what I'm going to key in on here are the processes of transcription and translation, so that when you go to read your lesson, you'll have a good feel, because we've already gone through, we've looked at some pictures together, and so I think it'll help as you actually do your reading after we get finished going through this together today. So the very first thing that we want to do is look at where these processes take place, and they actually take place within a cell. And part of it, one of them takes place within the nucleus. So here's the nucleus of a cell there and there. These are two different renditions by two different authors. This one shows more of the organelles that we're normally used to seeing within the cell, including like mitochondria, the endoplasmic reticulum. This author here has, since he's keen in on just the ribosome organelle, he has left the others out. And for our purposes, I thought that was good, because primarily that's what we're going to be dealing with, is the nucleus, the DNA, RNA, and then the ribosome. So I like this rendition here for our purposes, but I wanted you to be able to, see and remember that there are certainly other organelles that are within the cytoplasm, which is all out here, floating around in that cytoplasm. So let's see. I don't think there's anything else there that we need to. Oh yeah, one other thing here. One thing I do like about this drawing is that this author here has shown the DNA that's in the nucleus, and it looks like the information coded on the DNA is going to somehow be transported and put on RNA, and that it looks like the RNA is then what carries that message outside to the cytoplasm, out from the nucleus to the ribosome, where the protein is actually made. So kind of keep all that in the back of your head as we go to that first process that we're going to talk about, of transcription. Now here's a DNA molecule. Here's a DNA molecule. Remember, it's a two-stranded molecule, and the two strands are connected together by bonds between the bases. Remember, there are the four bases that you will find in the DNA molecule: the A, T, G, and C. The A always binds with a T, the G with a C. And this drawing here I like, because there's the one strand of of the DNA molecule, and the red is the other strand. And here you can see the actual pairing up. There's a G with a C and an A with a T, C with a G, G with a C. They have to be paired up appropriately. But what also I like about this picture is that it kind of shows what's happening in the process of transcription pretty well. Remember, we're going to take that DNA molecule, and somehow we have to get the information on some molecule that will then take that information out from the nucleus into the cytoplasm, where the ribosomes are, such that that code can then be used to make proteins. So what happens is that the DNA molecule, the two strands, actually split apart. You can see that happening here. And RNA is going to be made by using one of the strands of the DNA molecule. And what happens, if you look here, bases will come in, appropriate bases will come in, and they're very much the same bases that we see in DNA, the the AG and C. The only one that's different, rather than having the base T, we will have the base U. So wherever you think maybe a T would have been on the RNA, it will be a base U, uracil, instead. So here come these bases. They're going to match up. They're going to pair up with the appropriate bases that are on one strand of the DNA. So here comes the C, and it's going to bind with the G there. The G has to have a C. The A would have to have not the T, but a U. So here comes, we have a C and a U. The T on the DNA molecule would have to pull in an A. The C would have to match up with a G. And as we match up these bases, what we're forming here is actually an RNA molecule. And so the RNA molecule that is formed here is we're actually going to call it messenger RNA, and we'll talk about why in just a second. But it's this molecule then that is going to go from the inside of the nucleus, outside of the nucleus, carrying the code to make proteins. So here you can see the messenger RNA, those bases are matching up, with the appropriate bases on the DNA molecule. We're forming the messenger RNA. Once that is formed, the messenger RNA is free to move outside of the nucleus, and the two strands of the DNA molecule then go back together. So now we've taken the code from the DNA and we've put it on to a RNA, a very specific type of RNA, messenger RNA, that will now carry the message to the outside of the nucleus. Here, this is just a different picture of it. Transcription again. The blue is you can see the two strands of the DNA that are intertwined. They have to split apart. The bases come in, the appropriate bases come in and match up with the appropriate bases on the DNA strand, and in doing so we're forming the RNA molecule here, or RNA strand, messenger RNA, and then that messenger RNA can then take off and head outside of the nucleus, carrying the code in its own form, and then the DNA two strands can go back together again. So transcription is the first step of gene expression in which a particular segment of DNA is copied onto the RNA, and that new strand is called, we call it with a little m RNA. That's messenger RNA. So the messenger RNA is carrying, or acting like a messenger, it's carrying a message. So it's carrying that message or code from the nucleus out into the cytoplasm of the cell. So once that happens, now the code is now out into the cytoplasm, where we can now use that code to make proteins. And that involves the next process we're going to be talking about, which is translation. Now here's that messenger RNA strand right there, see? And remember, the messenger RNA strand that's been made, remember we brought in, it has the appropriate bases that came in and had matched to that one strand of DNA. So what's kind of interesting about this is that every three bases here codes for, it kind of has the code for the appropriate amino acid that it wants to come in and add to a growing chain of amino acids. So if you look here, the UCG, if we look down here, let's see, UCG, let's see. Some charts, they'll give you, they'll tell you the amino acids based on this. Some do it based on this. So the UCG here for the SER would be UCG right here. The UCG tells me that that is coding for the amino acid SER, if you'd like to call it that, or the real name is serine. So this is saying that at this particular place in our chain of growing amino acids, we want to have the amino acid serine. Now the amino acid serine also has three bases that are attached to it. And for it to be drawn into this particular spot in our growing chain of amino acids, these bases are going to have to match up to these. And let's see if they would. The U with the A, yeah, that would go there. C goes with a G, G goes with a C. So that would fit. So this molecule right here can match perfectly right up in here, and that shows that serine then will be added on to our growing chain of amino acids right at this point. Now if we look at the previous three bases here on the messenger RNA, the CGA, we have to match up with something that will then bring in the appropriate amino acid. Well, if it was the AGC, that wouldn't fit up there, so we wouldn't be bringing serine in there. So it has to be, the amino acid we brought in has to have the three bases attached to it that will fit right up with that C, G, and A. Let's see if it does. So the G goes with C, G goes with C, A goes with, remember in RNA, if you match an A with something, we match it with the base U, or uracil, not a T. So these particular three bases will match with a GCU, and the GCU is bringing right into this spot on the growing chain the ARG amino acid, or arginine. So obviously this wouldn't fit over here, this wouldn't have fit over here. This is coded specifically to bring in the correct amino acid to add on to the appropriate in the appropriate place to the growing chain of amino acids. And when you are making that chain of amino acids, what you're doing is you're making a protein. And remember that proteins are what decide the form, the function of everything that goes on in a living cell. It will tell that cell whether it's to become bone, if it's to become hair, if it's to become muscle. So, a lot is involved in getting the right coding here and then making the right protein to do what should be done in that cell. And remember, all of this started way back in the nucleus, remember, where the DNA transferred the information to the messenger RNA. The messenger RNA then brought it from the nucleus out into the cytoplasm. Here's that messenger RNA that has all those bases attached. Depending on what the sequence is of those bases, we will then be drawing in the appropriate amino acids to make a growing amino acid chain, which is then producing a protein. It says here in translation, messenger, just the same thing I think I've just said, messenger RNA produced by transcription from the DNA is decoded by that ribosome. Remember that messenger RNA comes up and attaches to that ribosome because it's the ribosome that's going to actually translate the code to produce a specific amino acid chain, and then after that amino acid chain will be formulated into a protein. So this next picture, all it really does is show the same thing. What's kind of interesting, remember we had the messenger RNA down here that have the bases on it, and then we had another thing that came in, bringing in an appropriate the bases that will match. Remember, and that thing was carrying a specific amino acid. Well, we actually call that thing a different form of RNA. It's not called messenger RNA, it's called tRNA or transfer RNA. It's transferring in some very specific amino acids dependent on what the bases are down here, and if the bases then can fit right into and match those bases here. So here we see that here's that thing or transfer RNA that has a specific amino acid on it. The three bases on it match to these bases, and so that brought the appropriate amino acid in here and joined it to the growing chain of amino acids. This one here, it looks like, here's our ribosome in the back. It's reading the code here, and it can read that code, and it knows what and we call that a codon, and this code up here on the transfer RNA, or this thing here, we call an anticodon. So we know what the codon here, or the appropriate three bases are here, and only the right bases can come in and bond onto these or attach onto these. So if this does come down and bond on here, it's telling us that the next appropriate amino acid would be the amino acid that these bases are dragging along in back of it. So it attaches on here, and then this amino acid will now add on to the growing chain. So if you look from picture to picture, here you can see that this last one we talked about, that last amino acid has come down, it's attached onto the growing chain, and then when we're done with these things or transfer RNA, we don't need them anymore, so they can actually be thrown off. And then we have spots for additional transfer RNA to come in, and match up here, if they do, carrying the appropriate amino acid that needs to be next in line of amino acids. So that's the process of translation. I hope that this helps as you go into your reading in your next lesson. You might want to refer back to this after you read your lesson. It would even be helpful to go back and watch this short video again. I think it would even reinforce and help you understand it even better. So thanks for watching, and I hope that this helps. Bye-bye.